new entity of cancer stem cell who actually are the majority of circulating tumor cells. We know that those cells are generally the progenitor of cancers, and they behave like progenitors for any mass that can be developed. We talk about a small proportion of the total tumor. Generally, they are highly resistant because they efflux all the xenobiotic outside the cell. Normally, they are slow-growth cells, but if they are triggered or they are in the perfect environment, they can grow exponentially, as I explained to you before. Nowadays, there are research to find out, or at least to detect, the circulated tumor cells which they have cancer stem cell features, and one mark is prominent, the CD133, and the other is the loss of OCT4 or 24 negatives, 2044 CD44 positives. This phenotype is applicable to breast cancer mainly. Of course, there are others like c-KIT, etc. The features of cancer stem cell is very slow, as I said, in a bulk tumor, exponentially grow when they engraft it in distant organ, and they perform what we call MET, mesenchymal to epithelial transition, and extremely high expression of MDR, multi-drug resistant pumps, or ABC, as we know them now. And they actually efflux all the substances outside. So, how we can detect those cells? What exists in the market? So, first we can isolate them by magnetic beads, actually immunomagnetic beads, who actually identified antigens in the membrane, up on the membrane. And in the backside of the antibody there is a polymer who includes iron. And when you put it in magnetic fields, it drags the cells of your interest down. So, such an antigen is EpCAM for epithelial origin solid tumors, or others, many others. Also, we can quantify circulated tumor cells by real-time PCR using a specific marker, the gene of a specific antigen that expressed in a specific tumor, like cytokeratin 19 for breast carcinoma, or EpCAM again. But what are the disadvantages? First, we'll talk about positive selection of cells based on one or few membrane markers, which means that you lose all the other subpopulation of circulated tumor cells. Second, magnetic beads is an enrichment method. It means that what you subtract is not only your cancer cells, it's also other cells inside, lymphocytes or monocytes. So you have other cells inside your samples. Also, magnetic beads, there are two kinds, the 50 nanometers and the 250 nanometers. The big beads are very sensitive in magnetic field, so when they detect your cell, it can easily be removed. But when your cell is in a dense cell environment, 250 nanometers is very difficult to find it. On the contrary, the 50 nanometers can easily move inside in multiple cells, but it's very weak when you put them in a magnetic force. So there are positive, there are negative things that you have to calculate and take them in consider. Yep, next. And of course, it's impossible to perform further studies like expression profile. Lately there was a journal, and I think it was from Imperial College, which actually measure, I don't like to mention the company's name, but the magnetic bead assays, and they concluded that you may quantify, but definitely you are underestimate the number of actually circulated tumor cells, and definitely you cannot perform. It is a method. The isolation of circulated tumor cell generally is reflecting to clinical outcome, but still it needs further improvement of how you will isolate them. Concerning the real-time PCR, the qPCR, you can only quantify the cells. You have no more material to work further. No cells anymore, no cell grow, and your DNA has been wasted only to quantify one or few markers only. That's why we concluded mainly to flow cytometry, as I said before. Flow cytometer can give you the single cell analysis ability to isolate the cell of your wish, and also it can be flexible to perform positive and negative selection. So if you would like to detect circulated tumor cells based on one or few antigens, you can do that. You can say, I want CD133 positive cells, you can have them. But also you can perform